Hello, everyone. Okay, so this is instructions for lab for the ABC cycle this week. I did put our new remote schedule here for the daily attendance, just so that you have that to refer to so you know when to show up for your Google Meets. So you are going to do the daily attendance for lab and class, two separate ones. So here's the button that you're going to click for the lab attendance. And then we'll go down to our lab folder. We are on lab 14 already. Can you believe that many? Okay, so we're going down to lab 14, and this is for the chromatography lab. You're going to open this in Cami and also submit using the turn-in button. Here are your due dates. So if you are an A day, it's due on Friday. If you're the BD group, it's going to be due on Thursday, and if you're in the C group, it's due on Monday. This is a relatively short lab, and it's going to be somewhat of a review because it deals with paper chromatography, and we've learned about it before. So you're going to open your chromatography assignment. Don't worry about the video yet, okay? It tells you in the lab activity when to watch the video. So you're going to open using Cami, using the Cami button, and I already have mine open, so I'm just going to click right to it. This is what you should see. It says Chromatography Lab, and here's a little introduction. It says the process of photosynthesis involves hundreds of substances and pigments like chlorophyll. Pigments give leaves color and capture light so the plant can make its own food. Three types of pigments in plants are chlorophyll, which is green in color, carotene, which is orange like a carrot, and xanthophylls, which are yellowish in color. We will attempt to separate and identify some of these substances by the process of chromatography. Chromatography is like a race between chemicals. The leaf pigments that dissolve the best in a liquid move the fastest along a strip of paper. The strip of paper is the racetrack, and the chemicals travel along the paper at different rates. You will race your chemicals up a paper strip to separate them and figure out which chemical is fastest and the winner of the race. The speed at which your pigments travel is based on how many hydrogen bonds the pigments make with the paper and the size of the pigments too. So unfortunately, we can't actually do this activity, but we're going to watch an Edpuzzle video based on this activity. So you cannot do this slide until you watch the Edpuzzle video. So what you should do is you should pause right now and then go back into Schoology, into the lab folder, and then complete your chromatography lab video, okay? And then you're going to come back into Cami, and you may even want to split the screen and have this open at the same time because you're going to draw in and label the colored bands to show how the results look from the experiment. So they're going to set up this chromatography strip. They're going to put pigment on here, and then you're going to actually draw in the colors. So you could either use the drawing tool. Just make sure you make it a thicker stroke thickness, and then make sure you're picking the right colors too, please. You can use I don't know if I would use the shapes tool, maybe the highlighter tool, the box highlighter, and pick your colors that way. But you're gonna draw bands on here with the different colors, and I would like you to label. So you're using carotene A, carotene B, xanthophyll, chlorophyll. I want you to label them the way they do in the video. Okay. And then you've got some questions for reflection. So number one wants to know how many different colors were separated on your chromatogram or the paper strip. And you're going to name the colors using the pigment colors. Okay. Why do you think only pencil marking can be made on the paper but not ink? So when you're writing on this chromatography paper, why would you use pencil to label it and not ink? And then number three, based on your results of the discussion of paper chromatography, which pigment would you conclude is the most soluble, which means it moved the farthest on the paper? Which one is most soluble in the solvent? And then what would you predict about the rate of photosynthesis of a plant whose leaf extract showed you 2 instead of 4 or 5 pigments? So do you think the rate is higher, lower, and then explain why you think that is? And then you're going to use the turn-in button up here to submit it to Schoology. And then you're done with this. It's a quick quick lab, should not take you very long, probably just one of these short lab periods that we're using now. But like I said, the due dates are listed in the assignment for you, so make sure you're getting them done by your due dates, okay? And again, email me if you have any questions.